Greetings everyone, and today we'll be exploring the biochemistry of pulse, which is a fundamental vital science that reflects the complex molecular mechanisms of cardiovascular functions. And while some of us might have learned about pulse in anatomy or physiology courses that we audited or took full-time as a biochemist, or you were just a passerby that saw a medical practitioner taking this vital sign, it's important that we know the biochemical process that leads to the generation of pulse in our body and also how it is being regulated. Understanding this will kind of help us to appreciate how metabolic, hormonal, even our cellular processes get to integrate to be able to maintain the cardiovascular function within the body. I mean, how it gets to regulate the homeostatic control process. So starting with the basics, let's look at what pulse is. A pulse is usually referred to the rhythmic expansion or contraction of the arterial walls, and you're able to feel this at different part of the body. But what is really happening at the molecular level, what is controlling this, is what we're about to talk about. When the left ventricle contracts during systole, it leads to the ejection of blood from the aorta, and this then creates some sort of pressure that then travels as wave. As you can see in the video on my screen right now, the prayer travels through the arterial tree at some speed that is approximately about 4 to 12 meters per second, and sometimes the speed is faster than the actual blood flow. So the key to understanding this phenomenon is first knowing why this thing usually do happen, this flexibility. Now, the fundamental thing about the flexibility of the artery is based on the composition of the protein. The artery contains the protein elastin. It contains elastin fibers, and this protein are basically made up of polymers of glycine and protein. And if you're familiar with the structural component of protein, of glycine, you will know that glycine is a very simple amino acid. The R group is not heavy. The same with proline, which is not the normal amino acid. It has some structural defect that is used or is responsible or helps in the recoiling or the elasticity of the protein that contains it more. So when prayer wave hits, the elastic artery is able to stretch, accommodating more blood, allowing them to flow through. And then during the diastole, there is a recoil. So that's what the expansion and contraction mechanism is all about. This elastic property of the artery is what is responsible for the pulse that we usually feel when we tend to maybe take a pulse. Sometimes when you put your finger close to your wrist, you're able to feel some of those pulse. So again, let me just try to run you through this video again. You see that wave moving through, and then it leads to expansion, and blood is able to flow through. That's what you then feel as prayer. Now, moving ahead, where can we really assess some of this pulse? There are 9 major parts of the body where we can get this pulse, and each of them have their own clinical significance. We have the upper part of the body, we have the lower part of the body, but I'll take them based on the way they appear. Now let's look at the upper part of the body, which has to do, starting with the head, the temporal pulse. The temporal pulse is at the temple. When you touch close to your head, you're able to feel that boom, 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 boom. Yes, where we have the superficial temporal artery runs through that place, and so it's very useful when other pulse points in the body are not accessible, or during cranial procedure. Now, the next one I'll talk about is the carotid pulse. This is the strongest because it's located at the neck and it's very close to the heart. So it's usually the primary site during emergency. You see when someone is lying down, maybe lifeless, and the first thing the medical practitioner does is to put hands in that place to feel the cardiac output that is still coming out, if it is weak or not even present at all. So in a situation like cardiac arrest, the first thing they try to do is to check how weak or how strong or the carotid pulse is. The apical pulse is also very unique in the sense that it's close to where the heartbeat itself is coming from. The apex of the heart is like the most accurate measure of the heart rate, especially in children, because this is closer to the heart where the pumping is really coming from. And then moving to the upper extremities of the body, that is talking about where we have the brachial pulse. That is where we have the hand close to the elbow. Whenever you touch that joint there, that thing close to your elbow, you feel that's blood pressure. Then also talk about the radial pulse, and this is around the wrist. This is very common. A lot of us have done that before at one point or the other, consciously or unconsciously, and even sometimes we even get to be examined, taking vitals in the clinics. Through that, it's very accessible and easy to check. Then I'll talk about the femoral. The femoral pulse is around the groin area where the artery is large, and it's used to assess I think most of us have not checked this before, but most times it's used to assess a circulation to the lower limb. The next one we're talking about is the popliteal pulse behind the knee. It's very difficult to assess, actually, just somewhat the same as the femoral pulse, which we don't usually pay attention to. The popliteal pulse is behind the knee, and it's kind of hard to find, but it is very important in evaluating the circulation to the leg. And finally, I'll talk about the posterior tibial, which is around the ankle. Right inside the ankle, you're able to feel the pulse in there. And the last but not the least is the dorsalis pedis, which is the pulse on top of the feet. It is able to assess the blood flow to our feet. And looking at it from a biochemical standpoint, I would say that the presence or absence of any pulse is able to tell us how well the vascular patency is. Sometimes it also reveals some material compliance from the heart and the integrity of the tissue perfusion. All of this may be determined or dependent on the endothelial function and also smooth muscle tone, as well as the metabolic status of. It's just more like a video that talks about the various more like a recap of all the pulse in the body. I'll just quickly run you through it. It's exploring the 9 parts of the body, starting from the upper part of the body, touching on the temporal, found in the temple, touching on the carotid, found on the neck. Then we go to the apical pulse that is found close to the heart, and we have the brachial pulse that is found behind the knee, and the radial that we can use our thumb to hold. I'm sorry, that's brachial wasn't knee, that was elbow. And the next, and which is the final one I would like to talk about, is the dorsalis pedis, which is on top of our foot. Yeah, so now let's look at the molecular details of how pulse is generated, and it all starts with the cardiac contraction through the process that is called excitation contraction coupling. You see just in the video when there is a synodal generation. I mean, the synodal node generates an action potential. Let me play the video again to let you see. When this action potential is generated, it leads to the depolarization of the cardiac myocytes, the membrane to become depolarized, and then this leads to the opening of the voltage-gated ligand, which also serve as the calcium channels in the sarcolemma. And then we have calcium ion influxing through this channel into the cell. When there is enough of this calcium ion within the cell, it triggers even the release of larger amounts of calcium within the cell, and this process is usually called the calcium-induced calcium release, where we have the sarcoplasmic reticulum releasing more calcium in response to a certain amount of calcium within the cell. This calcium within the cells then binds to a protein called troponin C, and the binding of calcium to troponin C causes a conformational change in the troponin tropomyosin complex. This is existent in the cell. The troponin tropomyosin complex is within the cell. When the troponin component of this complex binds to calcium, there is a conformational change that leads to the exposure of the myosin head within that tropomyosin. And when the myosin head is exposed, it becomes a binding site for actin filaments. The binding of actin filaments is usually motivated by HCP hydrolysis, where you have the ATP being hydrolyzed into ADP and CAMP. The binding of actin triggers the release of contractile force, which you now see as contraction thing that then leads to the injection of blood into the aorta. I hope I've been able to explain this excitation contraction. The excitation is as a result of the depolarization of the membrane, the calcium ion influx, and then this then couples with the contraction that results, contractile force generation that results from the binding of actin filament to the myosin head from the tropomyosin troponin complex. So I think I've been able to just break this down. I was just carried away. We have first the SA node depolarization, releasing action potential, then 2, the ATP hydrolysis, and this is usually called the myocardial contraction. Now, aside the contraction process, there is usually also vascular response, which is another mechanism that guides the pulse generation within the body. In this case, it involves the arterial wall, which contains elastin fiber, as I mentioned earlier, being responsible for the compliance and the smooth muscle itself. Now, when the endothelial cell, which is usually single layered, lining the blood vessel, when this endothelial cell produces nitric oxide, there is this. I think it produces nitric oxide from L-arginine. Yeah, produces nitric oxide from L-arginine, and this is usually catalyzed by nitric oxide synthase, if you're familiar with that cycle. I think some of this is also present in the urea cycle. Now, the nitric oxide diffuses through the smooth muscles within the cell, then activating the granulate cyclase, the enzyme that catalyzes the release of cyclic GMP. So I'm trying to connect that smooth muscle regulation of the vascular tone to the endothelial release of nitric oxide that then lead to vasodilation, and connect it with the cyclic GMP, which triggers the smooth muscle relaxation and vasodilation. Yeah, so the pulse prayer, which is usually the difference between the systolic and diastolic prayer, reflects both the force of ventricular contraction and the compliance of the arterial wall. And when there is stiffness within the arteries as a result of aging or atherosclerosis, there is increase in pulse prayer, and it's important that we know some of these things. I think I'll explain better as we move ahead, knowing fully well that the the pulse pressure is usually calculated as the systolic blood pressure minus the diastolic blood pressure. And it is important to know that the pulse is not constant; it's very dynamic, and it's dependent or regulated by multiple biochemical systems. So let's look at some of the things that could be responsible for what the state of pulse pressure would be. As I mentioned earlier, the force of ventricular contraction and compliance is what is responsible. It's what defines what the pulse pressure is. Autonomic system. Let's look at the sympathetic nervous system, which increases the heart rate through the norepinephrine. So how does it happen? When norepinephrine binds to beta-1 adrenergic receptor that is on the cardiac myocytes, if you're familiar with the G protein coupled receptor and how it works through some downstream signaling pathways, this is exactly what happens here. When the G protein coupled receptor that is present on the membrane is attacked, and it leads to the activation of adenylate cyclase. Adenylate cyclase phosphorylates or hydrolyzes, leading to the release of cyclic AMP. When cyclic AMP level increases, it activates an enzyme that is called protein kinase, which is basically responsible for phosphorylating the calcium channels. And this falls back to the previous mechanism that I explained, where you have calcium channels being opened up, increasing the influx of calcium. And when there is an influx of calcium, there is also an increase in the trigger, those other downstream trigger that leads to increase in the heart rate, as well as contractility. The expansion and the contraction then makes it to progress more because there's a trigger. So this is why sometimes when we are on a race or we are stressed, we have a higher pulse pressure. Now let us look at the parasympathetic nervous system, which does the opposite. In this case, when we have acetylcholine, an excitatory neurotransmitter from the vagus nerve, is able to bind to the M2 muscarinic receptor that is present on the surface of the cell, and then in this case there is a decrease in the cyclic AMP, which is in opposition to the other case in the sympathetic nervous system, where there is an increase in the cyclic AMP, and this is because the adenylate cyclase is deactivated. And instead of an influx of calcium, there is an efflux of potassium, which then obstructs the influx of calcium. In this case, there is a slow down heart rate. This slows down the process, the contraction, expansion contraction process, and then we have this regulation. The sympathetic is regulating the parasympathetic is regulating. So this is just like going the other way, and then they are both compensating for each other. And this is why when we take some deep breath, have a deep breathing, we have this vagal maneuvers. It helps us to slow down the pulse rates. Now let us look at what role hormones play in regulating the pulse rates. First, I'm talking about epinephrine from the adrenal medulla, which acts like norepinephrine to increase the heart rate. In the case of thyroid hormone too, we have the T3, T4 increasing the number of beta receptors, and when the number of beta receptors is increased, definitely we know that there will increase in metabolic rates, and this chronically elevates the heart rate. This is why sometimes when someone has hyperthyroid cases, there is a persistent tachycardia symptom. Someone with hyperthyroidism will definitely show some level of tachycardia when you have the pulse rate beyond 100. Looking at angiotensin 2, which causes vasoconstriction, increasing the peripheral resistance and blood pressure, and when we now have the arterial natriuretic peptide. Now, when I was preparing this, when I get to know about this arterial natriuretic peptide, which is a protein that is released in response from the heart's increase in blood fluid volume. Whenever there is an increase in the fluid volume in the heart, a protein is usually released in response to that, and that is natriuretic. This is released so that they can cause vasodilation and then lower the blood pressure. So when some of these one or two parameters are not well functional, that is when we start seeing a persistent blood pressure. But the body has a way of naturally regulating some of this pulse pressure as it progresses. Now, what is normal when it comes to pulse? Pulse varies significantly with age and also our fitness level. So, for example, when you see the table, looking at the table, you can see that in newborn we have 100 to 150 beats per minute, and also we have in infants like 3 to 24 months, we have 90 to 120. In children 1 to 10 years, we have 70 to 120. You see that in children generally the heart rates are very high, and this is because there are smaller sizes to their heart, which means that they need more frequent contractions to be able to maintain the cardiac output. But as we grow, the heart size are increasing, and then the metabolic rate per kilogram then decrease. So gradually there is decline in the pulse rates. So by adolescence and adulthood, the normal range falls to 60 to 100 beats per minute, and so we're able to get it maintained. Now, looking at people who are well-trained athletes who maintain some of these very high rigorous exercises per time, they stay within 40 to 50 BPM, and this is because not that something is wrong with them or is pathological. It's just as a result of some adaptive mechanism that the body has been able to put forward to be able to cope with those trainings or rigorous exercise that they engage in. So, in the parasympathetic tone and that they have as athletes, there is an increase in their parasympathetic tone, and this enhances the cardiac stroke. That's why sometimes when we get to exercise, it helps the heart to be used to that pumping stage. It's more like also keeping the heart fit. So when the heart doesn't need to beat as frequently to maintain that same cardiac output. Now, looking at the assessment of the pulse quality, I think that clinically we look at four major points to know how quality a pulse is. First is the rate, which talks about how fast it is, and the reading, which talks about how regular or irregular it is, and an irregular reading might indicate that there's an atrial fibrillation, or maybe there is a chaotic electrical signal that is capable of disrupting contraction, which is necessary for the pulse pressure to be maintained. We can also look at it in the sense of volume. What's the volume? How weak is it? How normal is it? Or how bounding is it? For example, a weak pulse may suggest that there is a poor cardiac output or an arterial occlusion. Something is obstructing it. A bounding pulse may mean that there is high stroke volume, and this may be something like an aortic regurgitation, that something is coming back, like a force that is an opposing force. So let me put it that way. And for people with hyperthyroidism, this can also show or display some bounding pulse type. We can also define pulse quality based on the character of the pulse, looking at the shape of the pulse wave that is being displayed. Just what I showed you the other time. Sometimes the way the wave moves indicates that there is a problem with the valve or there is a vascular disease. Now, when we look at the biochemistry of all this regulation or the generation of pulse itself, it's good that we look at the direct clinical implication of pulse abnormalities. Now, looking at tachycardia, I mentioned tachycardia the other time when I was talking about hyperthyroidism. A pulse that is over 100 has many biochemical causes. In hyperthyroidism, that means there is excess of the T3, T4, which means that this directly increases the heart rate by. Then I think how it does this is to upregulate the beta receptor. I mentioned that the other time. The beta receptor, adrenergic receptor on the membrane, and then this is where those things are able to bind with, which increases the influx of calcium and when leading to those other downstream effects of increasing the heart rate, and then further enhancing or increasing the cellular metabolism too. In fever, when someone has fever, there is also usually an increase in metabolic rate, and for every increase in our temperature that we see, there is usually an increase in pulse rates with around 10 beats per minute. So if your temperature rises from 35 to 37 to, that's like a normal body temperature, from 37 to like 39, that's like 2 degrees Celsius. That means you're looking at 20 beats per minute increase in your let's say you were on 80 before, we should be looking at 100 beats per minute. And because cells need more oxygen, this is one of the reasons why this pulse rate increases. In an anemic situation, when we have reduced oxygen carrying capacity within the blood, when we have less of hemoglobin that is able to bind to oxygen and take it to the heart, the heart pumps faster because there is need for it to quickly deliver adequate oxygen to the tissue. It has to be renewing the ones that are available. It has to quickly be pumping them and renewing them, and that's why in an anemic situation you also have a fast or high pulse rate. For bradycardia, let me move to that now. And before I leave that, let's talk about the catecholamine excess. When there is excess of some of these catecholamines, it also can reflect tachycardia, like acetylcholine. Now let me talk about the bradycardia. Bradycardia is more of hypothyroidism when you have less or lower level of the T3, T4. And also in situation where we have athletic training, as I mentioned before, where there is an adaptation of the heart to produce less so that it doesn't have to do that much work as it is being subjected to. So below 60 for someone who is non-athletic can be taken or indicated as hypothyroidism. Someone who is coming up with a pulse of less than 60 will indicate that person is hypothyroid patient, and so that means there is low level of the thyroid hormone, which has decreased metabolic rate and then have an implication on the heart rate. And when we have excessive vagal tone, perhaps as a result of medication or some certain medical condition, it can also slow down the heart rate. So I think key takeaways are that the arterial compliance depends on elastin and the flexibility of the muscle function, and also hormone is capable of modulating the vascular tone and cardiac output. I think this basically summarizes what I've been saying. And so I think it's good that we mention peripheral vascular disease. I don't know if you've heard of it before. This is more like the peripheral pulses help us to reveal what the vascular health status is. An absence of dorsalis pedis or posterior tibial pulse can tell us that there is arterial occlusion. So these two, when it is checked and it's not low or is not even present at all, shows that there is an arterial occlusion from atherosclerosis. And those plaques that contain some level of low density lipoproteins or some cholesterol crystals and calcium deposits that blocks the blood flow blocks the blood flow. So a weak femoral pulse usually indicates aortic disease, and when you have a bounding pulse like water hammer, then you know that this pulse is showing that there is an aortic regurgitation, more like an opposing obstruction, where the blood then kind of leak back into the left ventricle. This is somewhat serious, especially during the diastole. And in an hyperthyroidism state, as it may be due to an increased cardiac output that decrease the peripheral resistance. Now, as a biochemist, I want to look at this from a biochemist perspective. I think that pulse assessments generally help us to evaluate the cardiac output, especially looking at the autonomic function, the metabolic state, and the vascular integrity of some of this tissue, as well as determining the molecular processes involved. And all these are determined by the molecular processes that we discussed earlier.